Hello, welcome to pharmacy class. Today I am going to discuss in vitro and in vivo correlation. In vitro and in vivo data, that is known as IVIVC, are often used during pharmaceutical development. So for development of a pharmaceutical formulation, we have to perform so many in vitro and in vivo experiments. So in order to reduce development time and optimize the formulation, we go for the in vitro in vivo correlation. Correlation is a tool for predicting in vivo results based on the in vitro data. So that is the objective of the IVIVC. IVIVC allows dosage form optimization with the fewest possible trial in man and fixes dissolution acceptance. So that's why we go for the IVIVC. The IVIVC can be of three levels in which in vitro and in vivo data is matched. On the basis of these points, we can reduce the number of experiments. So simply, there are three levels A, B and C. So in A level, we match dissolution curve of in vitro with the absorption curve of in vivo. In B level, we match mean dissolution time with the mean residence time or mean absorption time it is. In level C, disintegration time, that is the time to have 10%, 50% or 90 or any one single point we are taking, and we can match with the Cmax or Tmax or rate of absorption, or similarly time to have 10%, 50% or 90% absorbed. So like that we can match simply single point matching. So here a correlation of this type is generally linear and represent a point-to-point relationship between in vitro dissolution and the in vivo input rates. Simply, in a linear correlation, the in vitro dissolution and in vivo inputs curve may be directly superimposable or may be made to be superimposable by the use of the scaling factor. So if the in vitro dissolution data and in vivo absorption data, that is plasma drug concentration time profile, matches point to point, that is superimposable, then we can say it is of level A IVIVC. Whatever the method used to establish a level A IVIVC, the model should predict the entire in vivo time course from the in vitro data. That is the outcome. When the level A IVIVC has been established, so whatever the method we used, we can predict in vivo data with the help of in vitro data. Level B IVIVC uses the principle of statistical movement analysis. That means the mean in vitro dissolution time is compared either to the mean residence time or the mean in vivo dissolution time. Here, level B correlation does not unequally reflect the actual in vivo plasma level curve, because a number of different in vivo curves will produce similar mean residence time. So that is the in vitro in vivo correlation. Level C. In level C, in vitro in vivo correlation establishes a single point relationship. So it is very simply matches the single point T50% in vitro method and at the similar point at T50% in absorption in case of in vivo. In level C, IVIVC establishes it is a single point relationship between a dissolution parameter. Simply, we take the T50% of in vitro dissolution data and T50% of the in vivo data and we matches. If it is matches, then level C in vitro in vivo correlation established. So level C correlation does not reflect the complete shape of the plasma concentration time curve, which is the critical factor that defines the performance of the product. Here, what is the significance of in vitro in vivo correlation? Level A correlation, which defines a linear relationship between in vitro and in vivo data, so that measure the in vitro dissolution rate along to determine the pharmacokinetic profile in vivo. After a proper validation, in vitro in vivo correlation predicts the in vivo bioavailability results from the in vitro dissolution data. It is very significant. Level A data, we can easily establish the relationship between the in vivo and in vitro, that is the superimposable curve, which reflect the in vivo behavior of the various formulation. So simply we are going to perform the in vitro test and we can predict the in vivo behavior of that particular formulation in case of correlation A. It is perfectly match, point to point matching. So here conclusion is that level A in vitro in vivo correlations define the relationship between an in vitro dissolution curve and in vivo input. Level A correlation should always be tried a priority in order to have a tool that allows a complete in vivo prediction from an in vitro dissolution curve and thus accelerate the development and assist in some regulatory aspects. As in vivo data are now well standardized, the main effort must be directed to the in vitro data. Various apparatus and media should be tested and assessed in terms of their in vivo predictability. The user should always be aware of the limit of the method and of the confidence of its prediction.